Welcome. In this lecture, we will talk about digestive system in human beings. Now, when we talk about digestive system, the first thing that we understand is the food that we intake gets broken down. There are two important ways in which it is broken down. One is called as physical digestion. The second is called as chemical digestion. Physical digestion is where the food particles are physically broken down, and chemical digestion occurs through the action of enzyme that acts on the food material, and because of which the food particles break down into simpler forms. For example, protein would break down into amino acid, fats would break down into fatty acids and glycerol, starch would break down into simple sugars. So there are two important ways through which digestion actually takes place. Now, for this digestion to have or to accomplish, there are few important pathways. One is absorption. The second is assimilation. Let's understand the difference. Absorption is where the food that is digested through the physical and the chemical processes gets absorbed through the intestinal walls into the blood. Now, this is absorbed for glucose, fructose, amino acids into the blood, and then there is glycerol, fatty acid, which enter the lymph system. Now, when glycerol and fatty acids enter the lymph system, these are absorbed by the lymph vessel or the lacteals. Lacteals are also called as lymph vessels. These lacteals or lymph vessels are present in the small intestine, and they help in the process of absorption. Now, this breakdown, as we understood, is facilitated by enzymes. Now, these enzymes act as catalase. They speed up the process of digestion. Since they happen in the body, they are also called as biocatalysts. These biocatalysts do the work of metabolism. Metabolism includes two processes. One is the catabolic process, the other is the anabolic process. That is, catabolic means breakdown, anabolic means formation or coming together. So both of these require enzymes which catalyze the process of digestion. The last is assimilation. Assimilation is after the absorption, this food is utilized by the various organs. For example, glucose is utilized for the respiration purpose. The excess of glucose is converted into glycogen in liver. Similarly, amino acid is used for the synthesis of proteins, and it is used by the enzymes. Also, it is used for construction of various organelles, the cell organelles. The next is fatty acid and glycerol. These get converted into fats. If we talk about the process of digestion, it happens in the alimentary canal. It begins with the mouth, moves to the pharynx, esophagus, stomach, small intestine and large intestine. Within the small intestine, we have three important components: duodenum, jejunum and ileum. Within large intestine, it is rectum, colon, the cecum, colon and rectum. That's the order. So order is again important. Now, if we focus on the whole of the alimentary canal, it is somewhere around 9 meters long. It is muscular, coiled and structural. Through the esophagus, the food moves through the peristalsis movement, that is regular contraction of the muscles that take place. And for this digestion to take place smoothly, there are various digestive glands. Digestive glands, the common digestive glands are salivary glands, pancreas, liver are some of the major salivary glands that we would focus as we proceed. To begin with the whole process, it happens or it begins in the mouth. Now, in the mouth, the opening in the buccal cavity is the mouth. This is where the food is chewed. It is actually softened and mixed with saliva. Now, saliva comes through three pair of salivary glands which are present in the mouth. We'll talk about the digestive glands in a while. We'll first understand the process. So, in the mouth, you have the lips. The function of the lip is basically they are soft movable structure, and they are mainly meant for sipping and sucking. The next to it is the teeth. Teeth is the hardest non-bony structure which is present. They are partially embedded in the gums. The next is palate, hard palate, the front one, soft palate towards the upper side of the palate, and that's the back side has the soft palate. So palate actually separates the buccal cavity with the nasal chamber, and therefore the role of palate is important. The next is tongue. So tongue is basically a skeletal muscle, but the movement of the tongue and the taste that we perceive is through the tongue. It is a muscular structure which is attached to the floor of the buccal cavity. And then we do have various glands which actually secrete mucus. The idea of secretion of mucus gland is to lubricate the food. It keeps our tongue moist, lubricates the food, and also release the secretions from the buccal cavity through these salivary glands. So the three salivary glands are parotid, which is at the back or beneath the ears, submaxillary, which is at the palate, sublingual below the tongue. So three pairs of salivary glands. The idea of salivary gland is to release salivary amylase, which converts starch to simple sugar. Next to it, we have the pharynx. As you can see here, pharynx is a funnel-shaped structure behind the soft palate, and this is where the food and the air would cross. So it gives a continuous signal when to open the esophagus and when to open the windpipe. The opening of the esophagus is what is known as gullet. The opening of the larynx is known as glottis. Very, very important. Both start with G, but an extremely important question for your multiple choice purpose. Esophagus, the opening is gullet, which is the food pipe. Windpipe through the larynx, the opening is known as glottis. Then there is epiglottis. The opening of the trachea in the pharynx is guided by epiglottis. Epiglottis is also known as uvula. And this is where it actually opens up when the esophagus closes. So it's the food pipe and the windpipe, as simple as that to understand. Next to it, we move to the esophagus. So esophagus is the passage which is around 25 centimeter long. The food passes down through the peristalsis movement, which is the regular contraction movement, and the food is in the form of bolus as it moves down. The next comes is stomach. Now stomach is a J-shaped muscular bag which has gastric juices that are secreted. It also has hydrochloric acid. The function of hydrochloric acid is to kill the extra bacteria which is present as the food enters it. Now the food gets churned here, and the churned food is known as chyme. So the name again is important. In the stomach, the food which is churned is known as chyme, and there are two important sphincters which are present in the stomach. The sphincter which is present at the esophagus and the stomach is known as cardiac sphincter, that is at the opening of esophagus merging to the stomach. The next is stomach meeting the duodenum, and this is known as the pyrolic sphincter. So two important sphincter, cardiac sphincter and the pyrolic sphincter. Next to it is the small intestine. The small intestine, as you can see, has three components: jejunum, duodenum, jejunum, and ileum, which is the last component. Now this whole small intestine actually is around 6.25 meters long. The diameter is only 2.5 centimeter. In contrast to it, large intestine has a diameter of 6 centimeter and is around 1.5 meters to 1.8 meters in length. So the length of small intestine is large, but the diameter is small and therefore called as a small intestine. The length of large intestine is less, but the diameter is more. Now small intestine has villi on the surface. The idea is to increase the absorption as the food moves through. So the glucose, fructose gets absorbed through the villi. Absorption of food through villi is one of the major aspects or the major works of small intestine. Now in the small intestine there is Brenner gland and crypts of Lieberkühn which secrete succus entericus, and succus entericus helps in the digestion process. The complete digestion of protein, fats and sugars happen in small intestine. The small intestine, as I said, is divided into duodenum, jejunum and ileum. Ileum is around 3.5 meters long, 3.5 centimeters wide. It is the largest part of the small intestine. Jejunum is 2.5 meters long and 4 centimeters wide. It is narrower than duodenum. Duodenum is the most important part. It is C-shaped, only 25 centimeters long. So a very small part of it is duodenum. Duodenum is where bile and pancreatic juice enter in. So when we would talk about the digestive enzymes, we would focus on two the enzymes which are released in two components, duodenum and ileum, which are extremely important. So most of them released in duodenum and ileum. In duodenum, it is mainly bile and pancreatic juices. All the pancreatic juices we would understand in a while. And then in ileum, it is the breakdown of maltase, lactase, maltose, lactose, and so on. So duodenum is again important. It has the descending limb. The descending limb of the duodenum is important because at the descending limb of the duodenum is where the common bile duct and the pancreatic duct enter. So duodenum, the descending limb, is where the common bile duct and the pancreatic duct enter. The descending limb is very important feature that you must know for duodenum, where bile and pancreatic ducts enter in. The last one is the large intestine. Large intestine is lying in the abdominal and the pelvic cavity, and this is again divided into three: cecum, colon and rectum. Cecum is commonly seen in grass eating animals, the mammals, rudimentary in others, and this is where the ileocecal valve actually permits the food to move from small intestine to large intestine but does not allow it to move backwards. So it's a one-way movement that is guaranteed, and from the cecum arise a blind tube which is known as vermiform appendix. The next is colon. Colon is an inverted U-shaped tube. It has the ascending limb, the transverse limb, the descending limb, and the lower section is known as sigmoid. After that is rectum, which is around 20 centimeters long, and the tip of the rectum is the anus, which is guarded by the anal sphincter muscle, and through which the defecation or egestion process takes place. Now the glands used in digestive system are important. Salivary glands, as we said, 3 pairs of salivary glands are present: parotid behind the ears, beneath the ears, and behind the ears, and they are the parotid gland. The submaxillary or submandibular glands are inside the lower jaw. Sublingual is below the tongue. All of these release saliva. Saliva is slightly alkaline in nature. The pH is 7.2. It's very important to note here. Water, salt and mucin is released. Salivary amylase breaks the starch into simple sugars. The next is liver. Liver is one of the largest glands which is present. Reddish brown in color, has a weight of around 1.5 kgs. It is located on the right side. It is made up of hepatic cells and produces bile juice which is stored in gallbladder. So released by liver, stored in gallbladder is the bile juice, and this bile juice is later released in duodenum. As we know, in duodenum, two important things, bile juice and pancreatic juices, are released. The next is the gallbladder. So gallbladder, as I said, is where storage of bile takes place. It is transparent ovoid sac structure in one of the lobes of the liver. It receives the bile from the liver through the hepatic duct and stores it within the gallbladder and empties it into duodenum, the descending limb of the duodenum, through the common bile duct. Very important question. In the descending limb of the duodenum. The next is pancreas. Pancreas is a leaf-shaped structure which is pink in color. It is located within the loop of duodenum and it is called as a dual gland. It has exocrine and it acts as an exocrine as well as an endocrine gland. As an exocrine gland, it is formed of acini or pancreatic lobules, and these produce pancreatic juice which empties into duodenum, which we would understand in a while, and helps to digest sugar and protein. As an endocrine gland, it is formed of islets of Langerhans, and these islets of Langerhans are important because they produce what? They produce insulin and glucagon, which aims to regulate the sugar level present within the blood. Now coming on to the major enzymes within the buccal cavity, as we start with, we have the amylase which converts starch to sugar. Pharynx and esophagus have no enzymes which are present. There is no mechanical or chemical digestion of food that occurs. The next is stomach. In the stomach, the first one is hydrochloric acid, which actually breaks or kills the germs. It makes the food acidic, destroys the bacteria. One important function is it activates the inactive propepsin and prorenin. Propepsin, the inactivated propepsin becomes activated pepsin. The inactivated prorenin becomes activated renin. So that is the function of hydrochloric acid, extremely important. Now pepsin. The function is to break down protein into peptones. So protein not into amino acid but into peptones by pepsin. Renin is used to digest milk. Now the process of digestion of renin is simple. Renin combines with casein, which is the milk protein, to form paracasein. Paracasein combines with calcium and it forms calcium paracasinate. Now this calcium paracasinate combines with pepsin again and gives rise to peptones, protease and polypeptides. Clear? So renin combines with casein, which is the milk protein, to form paracasein. Paracasein combines with free calcium ion to form calcium paracasinate. Calcium paracasinate combines with pepsin to form peptone, protease and polypeptides. Then within the stomach is the gastric lipid. Gastric lipase. Gastric lipase, as the name suggests, the function is to hydrolyze fat into fatty acid and glycerol. Then we do have gastric mucin. The idea is to protect the mucous membrane of the stomach. In the large intestine, by and large, water and salt are absorbed by the lining. The remaining unprocessed food is pushed through the rectum and leaves out through the process of egestion. Small intestine is one of the most important things that we would discuss. The small intestine, two important ways through which enzymes are released: duodenum and ileum. Let's understand these one by one. Duodenum is a very important aspect that we must understand. The function of duodenum is very simple, where we would talk about some of the major features. That first of all, the chyme is produced, and in the descending limb of the duodenum, common bile duct and pancreatic duct empties in. So bile is released. Now this bile has no enzyme. Its idea is to neutralize the acidity, make the solution alkaline and emulsify the fats which are present. So the idea of this bile is very simple. So bile, actually, I repeat again, the role of bile is very important. Bile has no enzyme. It neutralizes acidity. It makes food alkaline and emulsifies the fat. The next is pancreatic juice. Under pancreatic juice, trypsin and chymotrypsin are present. They are secreted in the inactive forms as trypsinogen and chymotrypsinogen. They made active and then they help in digestion of protein to polypeptide. So their idea is to convert protein to polypeptide. The next is carboxypeptidase. Carboxypeptidase hydrolyze polypeptide, which is created or generated by trypsin and chymotrypsin, into tri or dipeptide. So we are breaking protein step by step. So from protein to polypeptide by trypsin and chymotrypsin. With the help of carboxypeptidase, we are breaking polypeptide into tripeptide and dipeptide. Now, pancreatic amylase. Amylase, wherever it comes, it is breaking down starch. So, pancreatic amylase is also known as amylosopin, and this breaks down the starch and glycogen into maltose. The next is pancreatic lipase. As the name suggests, lipase, so breaks down fat into fatty acid and glycerol. It is known as stepsin. Pancreatic nuclease actually has ribonuclease and deoxyribonuclease. They break down nucleic acid into nucleotide and nucleoside. So nucleic acid is broken down into nucleotide and nucleoside. The next is ileum. Ileum is secretes succus entericus, and ileum actually breaks down the various components very interestingly. So the breakdown under ileum is important. So ileum, the first breakdown is irippsin or peptidase. Irippsin or peptidase breaks down this polypeptide into amino acid. Okay, so what is important is till the time of duodenum, proteins are broken down into polypeptide, di or tripeptide, and then they are further broken down into amino acid by irippsin or peptidase, which is released in ileum. Maltase break down maltose into 2-glucose. Very important. Sucrase, which is also known as invertase, breaks down sucrose into monosaccharides, that is glucose plus fructose. So breaking down of what creates what is important. Maltose would be breaking down into 2-glucose. Sucrose would be breaking down into glucose and fructose. Lactose would break down into glucose plus galactose, and that would be by lactase. Then we have intestinal lipase, which would break down fat into fatty acid and glycerol. Enterokinase, which would break down the inactive trypsinogen into active trypsin. And nucleotidase and nucleosidase, which have been created by the pancreatic nuclease, breaking down of the nucleic acid. They further break down the nucleic acid into nitrogen base, pentose, hexose sugar, phosphate. So those are the things that are broken down by the action of nucleotidase and nucleosidase. So breaking down of each of these is important. I repeat, under duodenum it is bile and pancreatic juice. Under pancreatic juice we understand 6 important things: trypsin or chymotrypsin, carboxypeptidase, pancreatic amylase, pancreatic lipase, pancreatic nuclease, which is further the ribonuclease and the deoxyribonuclease, breaking down the nucleic acid into nucleotide and nucleoside. Under ileum, it is breaking of polypeptide into amino acid by ribsin or peptidase, maltase, sucrase, lactase, which is breaking down of maltose, sucrose and lactose. And then we have intestinal lipase for lipid, breakdown of fat, enterokinase, which is converting inactive trypsinogen into active trypsin, and nucleotidase and nucleosidase, which break down the nucleotide and nucleoside into nitrogen base, phosphate, pentose and hexose sugar. So that is important. Now, the most interesting thing for today's lecture is the various tests. How do we test starch? Use 1% glucose iodine solution. If it turns blue-black, it indicates presence of starch. For protein, we have 2 important tests. One is Millen test and other is Burette test. Millen test is when egg albumin is heated, it turns out to red-brown, indicating presence of protein. It is called as Millen test. The name and the colors are very important. Burette test, when protein is combined with sodium hydroxide or potassium hydroxide and copper sulfate, it would give a blue-purple color. So it indicates presence of protein. So 2 tests for protein, those are Millen test and Burette test. For fat, one is grease spot. The spot would turn as translucent, very simple because of the fat. The next is Sudan 3 dye. Sudan 3 dye is used with oil and it gives a dark red color, very important, extremely important. Now, for carbohydrate, 2 important tests, Benedict test and Fehling test. Benedict test uses Benedict solution. So with the Benedict solution, you use sugar, boil it. A red-orange precipitate is formed where glucose reduces cupric to cuprous, and that leads to a red-orange precipitate. I repeat again, for carbohydrate, Benedict solution and Fehling solutions are used. For protein, Millen test and Burette test. For carbohydrate, Benedict test or Fehling test, which uses Benedict solution and Fehling solution. Benedict solution combines with sugar. When boiled, gives a red-orange precipitate, indicating that the cupric has changed to cuprous. Failing solution again combines with sugar when boiled, gives brown, brick, red, yellow precipitate, and this reduces cupric sulfate to cuprous oxide, indicating that there is presence of simple reducing sugars. Now for complex non-reducing sugars, what is done? Sucrose is combined with hydrogen chloride and dilute potassium hydroxide with caustic soda, and now this what is created is applied with Benedict test and Failing test. So ultimately what you use is Benedict test and Failing test, but you simplify that complex sugar into simple reducing sugar. So complex non-reducing sugar is simplified by addition of hydrochloric acid, dilute NaOH and caustic soda, and then Benedict's test and Failing test is used. So I repeat and summarize again. For protein it is Millen test and Buret test. For sugars it is Benedict test and Failing test. Extremely important. For fat it is Sudan 3 test. The precipitate colors are again important for each of those. So this was a quick understanding of the human digestive system in summary. If you have any questions, any doubts, feel free to post those in the comment below. I'll be more than happy to take your queries for the same. Wish you very good luck for your coming exams.